Good morning, everybody. Welcome to the webinar. We're just gonna wait for more people to join, and then something weird happened to my screen share, so I'll restart it again. Hold on. Okay, we'll start in about 1 minute. We'll start by going through our intro slides at 8:01. We're going to go ahead and get started and go through our intro slides. So today is the last webinar of this academic year. I know some of you have been out of school or maybe you aren't associated with the university for a while, but at Stanford and other universities on the quarter system, we're actually still in school, but we will make up for it in September because we don't start till the end of September. Today we're thrilled to have with us Masaaki Kitajima from University of Tokyo. He is one of a bunch of Japanese scientists hosting the 8th annual International Society for Food and Environmental Virology conference next week. So some of you on the call may be going to that. I think it's going to be an amazing conference. It is midnight in Japan, so we're really happy that he has joined us. We are also hosting today Daniele Lantange from Tufts, and we're thrilled that she was willing to join the webinar as well. Just a few announcements before we start. First, Kevin, please feel free to interrupt if I don't capture this correctly, but we're going to be starting a Discord chat group. If you scan this QR code, you can join, and that's where students working in this space can collaborate and network with each other. Everybody's welcome. Anything to add, Kevin? No? Okay.
I think that's good. Yeah.
Okay. And then there's going to be another journal club discussion that you can join if you want to scan this QR code on Friday, June 21st. And everybody is welcome to join that as well. And then lastly, we are looking for a postdoc to join our RCN. So if you or anyone is interested that you know of, please email a CV and 2 references to Kyle Bibby, who's on the call but hidden behind his black screen, or you can scan this QR code. There he is. So please spread that widely. And then yes, we also have an email list, webinar nominations, and I just wanted to say if anyone on the call has any suggestions for speakers or themes for next academic year, please email me. And there's also a nomination form I will be starting to put together with the team of PIs at the RCN, the webinar schedule for next academic year. And we've been really happy with the amazing talks that you all have contributed over this past academic year. So now I will stop talking and Masaaki can go ahead and share his screen while he's getting that set up. A reminder that if you have questions, please put them in the Q&A and I will be moderating questions at the end of Masaaki's talk. And just remember, it's midnight in Japan. So thank you so much, Masaaki, for staying up so late to join us today. We really appreciate it.
Thank you for your kind introduction. So, good morning, everyone. I'm glad to see many familiar names. I'm Masaaki Kitajima. I'm from the University of Tokyo. I'll be talking about our ongoing efforts on wastewater surveillance at international airports in Japan. So first of all, let me introduce myself and our lab. actually, our lab at the University of Tokyo is dedicated to wastewater-based epidemiology, which is named International Wastewater-Based Epidemiology Lab. This was launched about 3 months ago on March 1st in this year. And our missions are advancing research in wastewater-based epidemiology, educating young professionals in this field, and social implementation of WBE in Japan and overseas, especially in Asia. So myself and Professor Hiroyuki Katayama, who is my boss and PhD advisor, are leading this laboratory. And we are collaborating our industry partners, Shionogi, which is one of the major pharmaceutical companies in Japan, and Shimazu Corporation. I guess you may know this company. This is a very famous instrument company for chemical analysis, especially. So our activities it's not limited to wastewater surveillance at wastewater treatment plant, but also wastewater surveillance at international airports. So today I'm going to be talking about more recent efforts on wastewater surveillance at airports in Japan. So the background is after the public health emergency of after the end of public health emergency of COVID-19, we have now increased international travelers, and because of the global warming or global boiling, we are facing the public health crisis. We are in risk of imported infectious diseases in many parts of the world. So the wastewater surveillance for aircrafts and terminal is now attracting attention as an early warning system to monitor imported infectious diseases. So we are now working on this project to detect viruses using various molecular methods to demonstrate the concept of wastewater surveillance at airports. We are targeting many pathogens, and I'd like to show some of the results in a few minutes. So before I start talking about wastewater surveillance at airports in Japan, let me introduce, or maybe you may know these efforts at in the United States. So CDC is now leading the program called Traveler-Based Genomic Surveillance, or TGS. So this is publicized on the website of CDC. But CDC is monitoring pathogens at 7 airports. And out of 7 airports, nasal swab sampling or random sampling for human clinical samples are conducted at 6 out of 7 airports. And at 3 airports, wastewater samples are collected. So one of the, I guess this is New York airports, individual aircraft are monitored for wastewater. So this is a device used in this program designed by Ginkgo Bioworks. And for the other 2 airports, probably this is in Boston, or and this may be San Francisco. But these 2 airports are triturator. So wastewater samples are collected from triturator, and viruses are being tested. So this is the situation in Japan. So in Japan, for travelers, voluntary testing is conducted only for symptomatic travelers. So this is a difference between Japan and United States in terms of testing for travelers. In the United States, random sampling for travelers is conducted, but in Japan, only voluntary testing for symptomatic travelers is conducted. So this clinical surveillance is conducted at 5 major international airports in Japan: Haneda, Narita, Kansai, Chubu, and Fukuoka. And this is the list of pathogens tested for travelers. So a variety, as you can see here, a variety of pathogens are tested, including SARS-CoV-2 and influenza and so on. So we are now proposing the concept of Japanese version of TGS. So by referring to CDC's TGS system. So we are now collecting aircraft wastewater samples from treatment plants dedicated to aircraft wastewater. And we are also collecting wastewater samples from terminals. And so our concept is to collaborate with quarantine so we can provide data from wastewater to the quarantine for more efficient surveillance by combining wastewater and human clinical sampling. And we are now collaborating, we have initiated discussion with CDS representatives for collaboration between our group in Japan and CDC's group. So so this is the Japan's TGS concept, combining quarantine and wastewater. So now you may wonder where should we collect wastewater samples from at the airports? There are several options. So first, as I mentioned in the CDC's case, we could collect wastewater samples directly from individual aircraft. Then the next option is we could collect wastewater samples from laboratory truck after collecting wastewater samples from a few aircraft. And then normally laboratory truck carries wastewater to treatment plant for aircraft wastewater within the airport. Another option is terminal wastewater. So normally we can collect terminal wastewater by opening manholes in the airport. And at some airports, there is a wastewater treatment plant in the airport. So some of the airports treats wastewater from aircraft and terminal at wastewater treatment plant. So as you can imagine, if you collect wastewater from aircraft, you can gain information more granular, but less comprehensive. If you collect wastewater sample from wastewater treatment plant, you can obtain comprehensive information, but less granular. Now, there are some challenges, limitations and challenges in collecting wastewater samples from individual aircraft. Let me list here. So it's impossible to definitely identify the original pathogens because there are transit passengers at every airline flight and carryover from previous flight because wastewater tank is not completely cleaned at the destination. And second is concern of country level conflicts, so which we have to avoid. For example, stigma for a certain country if we report pathogen being detected from a flight from a certain origin. Third, we need ethical considerations, especially privacy, and we need to obtain consent. But for this aspect, guidelines and laws are not developed. And fourth, not all passengers use toilet in aircraft. This is major limitations, especially for short flights for like a few hours. The fifth, need considerations from airline companies. So the challenges we are facing is that we have difficulties in obtaining permissions from airline because of a lot of concerns like negative reputations and impact on scheduled or on-time operations for them. Especially they have limited manpower and a lot of time and effort are required for sampling from individual aircraft. So for like ground handling stuff, so this is additional effort. So it's kind of difficult to obtain permissions from airline companies. And then because there are so many flights arriving in Japan, in Japan's case, there are 3,300 flights per day arriving to Japan. So it's almost impossible to collect this number of samples from all the arriving flights. So it's not necessarily efficient to collect wastewater samples from individual aircraft. So for the purpose of monitoring the import of pathogens in our country, we thought aircraft wastewater treatment plant and terminal wastewater may be more efficient for this purpose because of these limitations and challenges. So we collected, based on this background, we collected wastewater samples from airports, anonymous airport in Japan. At one airport, we collected wastewater samples from a treatment plant dedicated to aircraft wastewater and terminal wastewater like this. From terminal wastewater, we used passive sampling and grab sampling because the terminal wastewater fluctuates over time in terms of viral load. And at the other airports, we collected wastewater samples only from terminal. And for comparison, we also collected municipal wastewater from wastewater treatment plant in Sapporo City in Hokkaido in the same period of time when we collected samples from airports. So this is the method that we used to detect viruses from wastewater or epicence-based methods. So these are our original methods that are highly sensitive to detect viruses from wastewater. So we use this. So I'm gonna skip details, but after obtaining DNA and RNA extracts, we did qPCR, and we also did short-gun metagenomic analysis using NGS. So here, this is the results of the detection of pathogens or viruses from terminal wastewater. So I show here the comparison between passive sampling and grab sampling. As I expected, so passive sampling showed higher virus detection rate, which is not too surprising. And as expected, we validated that passive sampling is more comprehensive in terms of the detection rate. And because terminal wastewater, of course, has time fluctuation in terms of virus load. So here's the results of the pathogens that are tested for travelers. So that's why for these pathogens, we have the data on airport clinical surveillance. So in this period of time, 104 individuals were tested, and this number indicates how many people tested positive for each pathogen. As you can see here, influenza A and SARS-CoV-2 were detected from many travelers, symptomatic travelers. And from wastewater treatment plant, municipal wastewater treatment plant, a variety of pathogens were detected, as you may expect. And from terminal wastewater, we also detected a wide variety of pathogens. And yeah, if you compare airport A, terminal wastewater versus aircraft wastewater. Yeah, what I'd like to highlight here is that for some pathogens were detected only from terminal wastewater. For example, in this case, influenza B and rhinovirus were detected from terminal wastewater, but not detected from aircraft wastewater. But on the other hand, parainfluenza virus was only detected from aircraft wastewater. So this means that we probably need to test both terminal and aircraft wastewater for comprehensive pathogen detection. Another thing I like to draw your attention is that, more importantly, so we detected some pathogens that were not detected by clinical surveillance, but we did detect from in airport wastewater samples. This means that these pathogens were overlooked by clinical surveillance, but we can pick them up by wastewater surveillance. So this means that we demonstrated the usefulness of airport wastewater surveillance as a complementary test to quarantine. So this is the results for the pathogens that are not tested for travelers. That's why we don't have clinical surveillance data. But for these pathogens, gastroenteritis viruses, as you may expect, we detected from municipal wastewater samples with high frequency. And we also detected them from airport wastewater samples with high frequency. And we tested actually herpes viruses as well, EB virus and cytomegalovirus. We also detected them from municipal wastewater samples and aircraft wastewater samples. Yeah, so these viruses are like commonly detected from wastewater samples, but this demonstrates that wastewater surveillance can widely detect viruses, including the viruses that are not tested by clinical pathogen surveillance. So yeah, this is the last results. So we did shotgun metagenomic analysis for the same sample set, and we detected like SARS-CoV-2 influenza by qPCR with high frequency. And we also found this one, these viruses with high abundance with metagenomic sequencing. And I focused on rarely detected pathogens by clinical testing. For these pathogens, we also detected by metagenomic sequencing. So yeah, specifically these pathogens, parainfluenza, common cold coronavirus, and paracoviruses. So this these pathogens were also detected by qPCR, as I shown in the previous slide. So this validates the results of metagenomics, and this demonstrates the usefulness of comprehensive pathogen surveillance using metagenomics. So this is a summary. We did the proof of concept study, and this has demonstrated the potential usefulness of airport wastewater surveillance in Japan. And for in future, this wastewater surveillance can be a valuable tool for efficiently monitoring imported pathogens, including emerging and re-emerging ones, and tropical pathogens. So with that, I like to finish my talk, and this just advertisement. Our laboratory welcomes international collaborations, especially from the US. So if you are interested in working with us, please email me at this email address. And another thing is, in addition to our RCN postdoc, there is another position available in our lab. So if you are interested in working with us, then please scan this QR code and apply. Thank you very much, and I'll be happy to take any questions.
Great. Thank you so much, Masaaki. Great talk. Just a reminder for all the attendees to enter any questions you have. There's several questions here into the Q&A box. And you can also upvote them if you are interested in some of the questions that have already been answered or entered, excuse me. So with that, I'll get into a question here. Can you expand more on the ethical considerations of sampling and what that might imply at different sort of sampling scales at the airplane or further downstream?
Yeah. Thank you. Yes. Unlike municipal wastewater plants, the target population is much smaller, like hundreds of people. So if we detect a certain pathogen from a certain individual aircraft, that means we can target like a few hundred people. So for that aspect, we need to protect their privacy. Of course, if we publicize the data, we gotta be careful, but at least we can know the presence of certain pathogen from such kind of small community. So we have to consider that the privacy aspect. And another thing is consent. We don't obtain consent to test wastewater samples for such small population. So we have to consider such kind of consent and personal information for So that's what I meant. Does this answer your question?
Yeah, I think it does. I think it's something a lot of us have been thinking about and sort of at different scales, and definitely with this falls into that realm. Another question was, are these pathogens shed via urine, feces, both? We don't know. The likelihood of detection given a case on board or something like along those lines.
Right. Yeah, some pathogens are excreted only feces, and some pathogens, relatively smaller number of pathogens are excreted only in urine, and some pathogens are excreted in both. But yes, since we are targeting toilet wastewater, basically, I believe we can target pathogens excreted from both urine and feces. I think this is, but another limitation is aircraft wastewater doesn't contain much amount of saliva, unlike municipal wastewater. So that's probably one of the reasons why we observe the different populations or composition of viruses from aircraft and municipal wastewater. If we look at details of results, that's probably the reason. Yes.
Great. Another question here from Colleen Naughton is, what was the turnaround time for sequencing? And I'll expand that maybe just for your whole pipeline in terms of decision making if you were to detect an emergent pathogen, for example.
Right. So we are in still a research phase, so we haven't implemented yet. But turnaround time could be maybe a few days. If we routinely do the sequencing analysis. But maybe could be shorter than that. But since we are using standard metagenomic sequencing tool, I think currently maybe a few days. But in future, definitely it's important to shorten the turnaround time, like or the next day or something. Yeah.
Great. And I'm going to try to get to as many of these as we can in the next 2 minutes or so. Allie asked in the chat, it was very interesting that you observed the causative agent of whooping cough. Do you know if were cases of whooping cough in Japan or in that area? And do you think that your passive sampler works well for bacterial targets?
Yes, this one, right?
Yeah.
Paratusis, right. Yes, in Japan, not so many cases are reported for this. So I was also very interested in these results. Yeah. So yeah, our passive sampler and our absence method does work for for bacterial pathogens and bacterial DNA as well. Yeah, we have validated this too. Yes.
Great.
So
And I'll get to one more, and then apologies if I wasn't able to get to your question. And since it's midnight where Masaaki is, maybe if you're interested, you could follow up with him over email. I think he's going to take off after this talk. But did you observe correlations, or do you think there's correlations? Were you able to determine between your observations in the wastewater and clinical tests, or were there not enough clinical tests to do that? And did you account for visitors to the terminal, for example, people working there that weren't in fact traveling?
yeah. I think this slide answers your question, I guess. All we have for clinical data is this. So this is publicized from Ministry of Health and Labor in Japan. But so this is the data for clinical side. As you can see, for like SARS-CoV-2 and influenza detected from both. But as I mentioned, some pathogens were not detected from human clinical samples, but they were detected from wastewater samples. This is somewhat different, but so like somewhat agreement, but somewhat different. This may be the value, or maybe the value of wastewater surveillance.
Great. Well, thank you so much, Masaaki, for a great talk. Very interesting work. And like I said, apologies if I was not able to get to everyone's question. So with that, we'll go on to our next talk, Daniele Lantange from Tufts University. Take it away, Danielle.
Hi everyone, and can you hear me? Is everything okay?
Yes, we can hear you.
Thank you so much. So a little bit different presentation and I think really complements. I'll be talking today about expanding wastewater surveillance to low and middle income countries, example from waste effluent surveillance in Côte d'Ivoire and Liberia. The project is a large consortium of people. It's the Stop Spillover Project funded by USAID. Spillover as a project has 3 different goals: to strengthen capacity in priority countries to monitor, analyze, and characterize risks of priority zoonotic viruses, to develop, test, and implement interventions to reduce the risk of spillover, and if spillover occurs, to mitigate the amplification and spread of priority diseases. The work that I'll be talking about today falls into this first category: monitor, analyze, and characterize. So just to say, we'll be talking today about projects in Côte d'Ivoire and Liberia. They're both countries in West Africa. They border one another. You can see Côte d'Ivoire there, and Liberia is the one in gray. That intersection of Liberia, Guinea-Conakry, and Sierra Leone is where the 2014 Ebola outbreak in West Africa started. So the priority pathogens in Côte d'Ivoire and Liberia are Lassa, Ebola, and highly pathogenic avian influenza. And a lot of the work in these countries is building on work that started after the West African Ebola outbreak. The What I'm helping on the project is on their surveillance, so working on rodent sampling, surface sampling in households with rodents, with Lhasa, and on their wastewater sampling. And on another set of slides, just as a teaser, we have sampled households with rodents that are identified with Lhasa, and also seen Lhasa virus, we don't know if it's infectious or not, on surfaces in those households. So we can swab household surfaces to kind of look at rodent and Lhasa impacts. But today I'll be talking about our wastewater surveillance. I think we've all seen graphs like this. We know, especially in this group, that worldwide expansion of wastewater testing happened in SARS-CoV-2, and many high-income countries have beautiful graphics like this. This is the one from Boston, where I live. What people might not know is we did wastewater sampling for polio, a low prevalence virus, as part of the certification of eradication of polio in a country. So for 3 to 5 years, if you did not see polio in wastewater, which was mostly in urban areas of countries, it was part of the certification to become polio-free. Now, most of the sampling, polio and SARS-CoV-2, is in sewer systems, sampling with automatic samplers for composite samplers or grab sampling. We all know this. But the limitations of this are massive. 43% of the world does not have safely managed sanitation, does not have access to sewers or wastewater treatment plants. And there's very little testing outside of these sewer networks in wastewater sampling. There's some papers that are essentially kind of rivers that act as sewers. And there's almost no testing that I've identified, and I know people are working on this, maybe people on this call, looking at wastewater sampling in sludge, fecal sludge, looking at latrines or fecal sludge management. And so there's opportunities for expansion of wastewater sampling to these underserved populations. Now, I'm on a spillover project, and of course the donor wanted wastewater sampling and everybody was very excited about it. But when I was asked to help them on this, there's an inherent tension. By definition, spillover risk from animals to humans is not highest in areas with sewage systems. And animals don't conveniently poo into a sewer system for you. And the concept of wastewater for sampling for surveillance for spillover is novel. And so we really wanted to start in this project with our highest potential for success. So we wanted to take a high pathogen concentration with a known successful media. One of the things is for a lot of these pathogens, there isn't even the laboratory data to know, do they survive in urine? Do they survive in wastewater? Do they survive in fecal sludge? Do they survive in water? With an appropriate sampling method. And we thought that if we brought those three together, we'd really have a high potential for success in using waste effluent surveillance in these contexts. So our process was we reviewed the research for each of our pathogens. We identified our likely places of highest virus prevalence in the environment. We identified their media to be tested, the exact testing locations, determined an appropriate sampling method, safely collected samples and transported them to the laboratory, and ensured appropriate testing and sampling to kind of pull all this together. So I'm going to start with our work in Côte d'Ivoire and who will test what where. So our first location was open small canals that contain human wastewater downstream of slums in the urban area of Abidjan. We were going to test for, this should be SARS-CoV-2, the pathogen of SARS-CoV-2. There's a lab in country that was IPC that already was testing for SARS-CoV-2. And so we knew they could safely manage this pathogen. And we were going to do active and passive sampling with tampon Moore swabs. Now, our second sampling site was open, very small, intermittent waste streams that contain chicken slaughtering liquid waste in markets in Grand-Bassam. Grand-Bassam is another area of Côte d'Ivoire, and when they slaughter the chickens, they put the blood and materials into bowls, which they then dump into very small, maybe 3 to 6 inch waste streams that flow through the market. They're intermittent. It depends on when people are slaughtering the chickens. The pathogen we were looking here for was highly pathogenic avian influenza. The laboratory in country was Lanata. Again, Lanata was already testing for HPAI. So we know that they would be able to do this. And because it was intermittent streams, we looked at passive sampling. Now, our third place that we wanted to look for, and we're working hard on now, is testing at the main wastewater exit pipe of the district hospital in Mon. This is on the western side of Côte d'Ivoire on the Liberia border. We think there's a lot of Lassa there that's undiagnosed and treated as a fever, as malaria. And we're looking to do passive sampling at this wastewater exit pipe. But we're also, and I'll come back to this later, thinking very hard about safety and about these higher level BSL-4 pathogens and how to test them in-country labs, because it's a requirement of the donor and the country that samples do not leave the country. So I'm going to give some pictures. This is the testing of SARS-CoV-2. Here's the kind of wastewater streams coming out of the canals downstream of the slums. Here's IPC staff collecting samples with their grab sampling, which they had been doing on a different project. Here's putting in passive samplers, and here's collecting the passive sample. Here's Lanata testing HPAI. And in the end, I think we got avian influenza instead. It's kind of lab capacity what was able to be done. These are these very small chicken waste streams. We just grabbed a bench and put a tampon, an organic tampon, in the waste stream, and you can see these waste streams moving through. So actually, in our proof of concept, we met our goal of showing that we could measure these pathogens in these areas with the laboratories in Côte d'Ivoire. So we had a comparison. Our SARS-CoV-2 waste effluent passive samples are on the left. Our active samples are on the right. We had 13 of 24 of our active samples were positive. 9 of 24 of our passive samples were positive. That statistically comes out as a non-statistical difference. And our CT values were quite high. There's not a huge prevalence of virus here, of 28.2 to 35.1. The same results for HPIA. This actually ended up being avian influenza. Waste effluent samples, we had 3 positives in our in the chicken blood waste out of 24. Again, very high CT values, 29.3 to 32.4. So what are our successes? This is a large international collaboration showing that we can do this in proof of concept with various methods. We can identify some of these pathogens. It's low cost and highly effective. And it's a continued development to kind of democratize access to wastewater sampling outside of just high income countries that have sewer networks. Now, our challenges. First, laboratory supplies. Most of the supplies that were used from the PPE to the QIAGEN kits I brought in as checked baggage on flights. There's much more training and supplies needed for more quantitative PCR results, for sequencing. We're hoping to do sequencing. We're struggling with sequencing in country, and expansion to higher risk and spillover pathogens. This was a proof of concept. There's an incredible amount of interest and push from the donors to do wastewater surveillance, because it's a buzzword everyone's talking about, but how to actualize it in these countries with the lab safely is a real question we're working on and kind of struggling with on a day-to-day basis. Now, to come back to the Lassa isolation ward, this is a Lassa isolation ward in one of the largest hospitals in Liberia. You can see the writing here in the back. It talks about the isolation unit for Lassa patients. There's a pipe that comes out that's all the waste from the unit. In the foreground here at the bottom left is the septic tank for the Lassa waste, which flows through the ground, and just as a normal septic tank is how the treatment happens, right? So we'd like to move to higher risk pathogens. Lassa fever is one of them. In particular, Lassa has not yet been identified in waste effluent surveillance or wastewater in real world circumstances, although Kyle Bibby is doing work in the laboratory. And I think it's really important work to look at these kind of neglected viruses in the laboratory to understand what we can do. We need proof of concept that we can even get Lassa on a cotton tampon swab and be able to have the lab be able to analyze it. So we're starting here. When there's a Lassa patient, we're taking passive samples from an access point that we put into this drain pipe, and we know it should be there. So we're testing our testing methods in the lab. We are lucky in Liberia because of the Ebola outbreak. There's an incredible amount of trained personnel and trained laboratory personnel that remain that are very safe and comfortable working with high risk pathogens. And so we're very confident in our safety plan here. And we were hoping, we're on week 3 of sampling of 12 weeks. We were hoping to have results by today. We don't have results yet, but that's forthcoming. We also want to do Lassa sampling at the regional hospital, Mon in Côte d'Ivoire. This is the wastewater drain for the entire hospital. So the Mon Hospital is a rural hospital. It feeds the western area of Côte d'Ivoire. They are part of a larger project called What is Fever? When you present with fever in a hospital like this, it's often default treated as malaria. And then if the malaria meds don't work, you may or may not come back and may or may not be able to be tested for other types of fevers. And so there's likely quite a bit of undiagnosed Lassa in this hospital. So we'd like to test this waste, which now exits the hospital untreated into this forested area to see if there's Lassa there, as mostly see if we can test it, and also to see if we can advocate for better rapid test kits and testing in this hospital, because we think this is an emergent Lassa region because we've seen rodents with Lassa here. This is stalled at the moment, and I've been on calls. I understand my previous presenter with late night and early morning calls lately, because I've been on a lot of calls. We are working very hard to understand the safety of working with Lassa in a country that has laboratories that don't have the same experiences with Ebola, and how we can do that safely. And we're really working pretty hard on that right now. So what are our results? This proof of concept worked. We have proof of concept of sampling intermittent waste effluent streams in LMIC with LMIC laboratories doing capacity building to get some results. What next? We want to publish this. We'd like to get our sequences. We're working on that with the lab. There's one lab that can do sequencing. It's been a bit of a struggle. We're working on it. We'd like to move toward additional pathogens, particularly spillover pathogens. We'd like to see what we can do in more animal areas, like bat caves that have water underneath and streams that come out would be a prime sampling location for things like this, etc. One of the limitations of this work, and I think it's an important, I'm not critiquing, but it's an important one, is we can do no broad spectrum testing for viruses because we cannot identify a new virus under USAID. So we have to test for Lassa or for HPI only. We can't do broad spectrum sampling as part of USAID rules and I'm not disagreeing with these. They're just kind of as part of this project. We can't do broad spectrum open-ended viral testing and to disseminate this. So I wanted to summarize and just say there's a need to extend the benefits of wastewater surveillance to areas without safely managed sanitation. We develop methods to test waste effluent streams in Côte d'Ivoire and successfully use these methods to identify human and non-human animal-derived pathogens. And waste effluent surveillance is a promising technology for expanding the benefits of wastewater surveillance to LMIC and humanitarian contexts as well. Have a call next week with NSF to see if they can incorporate this into their facilities. So I'll stop the share and come back to the group. And I wanted to thank Kyle, who's been helping us with the tampon sampling very much and the Lassa work, and to Ellie and to everyone for having us and for being here today. So thank you.
Thank you so much, Danielle. You have been working incredibly hard on this project in a very difficult situation, and it's amazing to hear. And I know I would be incredibly frustrated with all of the hurdles that you have to jump through. And I wondered if you could sort of, I don't know, I think we're interested in hearing about some of those. So there's some questions related to that, which is, can you talk about one of the challenges, which is actually getting like instrumentation in country? How do you get like a PCR machine that's functioning well in country? Did you have to do that, or how are you handling that particular challenge?
Right. And I think this is that, I mean, Marlene's question is exactly, we are a little bit lucky because these countries, not that anything is lucky about the Ebola outbreak, but Liberia has been well stocked in Ebola and afterward with laboratories with PCR machines. So we do not actually need to bring in the machines themselves. But we are using just standard qPCR, right? This isn't digital PCR. We'd love to bring in a mini ion, right? We're also hampered in this project because it's USAID and we're not allowed to bring in equipment. So anything over $5,000 we can't bring in. So we end up partnering, like one of the labs in Côte d'Ivoire is working with FAO and is going to get a BSL-3 facility, right? And so once FAO builds the BSL-3 facility, we can then build on that. We are trying to do what we can safely with what we have. And I think you see we're adapting those methods. And it has been a ton of time for very small results. I think, Ali, that you're completely correct. And there are times I would like to tear my hair out. I will be totally honest about that, about something. And it is this sense of, yes, this would have been infinitely easier if we just sent all these samples to Germany or Italy, right? Mm-hmm. But there's a question of what's our responsibility to build capacity and what is, so in some degrees, an outcome of this is not just the samples and the results, but the capacity building, right? And the training. And we can bring in anything that's under $5,000. So I regularly travel with 4 suitcases of 50 pounds each with, many, many thousands of equipment, many, many thousands of dollars of equipment that I'm bringing in. Right. But I do think it's for the larger digital PCR sequencing. I think you're depending on your larger donors through other grants, like you're building off of other grants that can do that type of laboratory infrastructure. Right. I don't know if that answers it.
Yeah, I mean, thank you for addressing that question. There's lots more questions about, like, on this subject, but maybe just to change the subject a little bit. Can you tell the audience who are less familiar with Lassa, like why it is a high priority pathogen, and then maybe discuss how you chose your list of pathogens and if it would be possible to maybe add easier targets there, things that you might culture, for example, that would be easier to start getting data for sooner. Sort of talk about that a bit.
Yeah, and this is the whole tension. So, and this comes back a little bit to donors and funding. So the whole set of projects is supposed to be spillover, and the high-risk pathogens were chosen in combination with country governments. So at the outset of the project, the country governments chose the pathogens they wanted to work with. That involves numerous partners, the governments and the donor together. Those pathogens depend on what they think we'll find in the area. Lassa is transmitted from rodents, rats, mostly mastomus, not completely, and is a fever with a very high morbidity and mortality. We don't tend, I mean, it's similar to Ebola, honestly, although it hasn't caused as large of outbreaks as of yet. And so there's quite a bit, there's a lot of undiagnosed Lassa. There's a lot of Lassa that is, it has a higher rate of not having any of asymptomatic. And so we think there's just a lot of circulating Lassa that we don't know about that could then bubble up, right? So this is actually the inherent tension of this entire project, which is what I'm kind of managing. Is I came in year 3 of a 5-year project. Everybody said they wanted wastewater surveillance. They read all these wonderful papers about how great it was. And they said, we're going to do wastewater surveillance. And it takes someone who knows, like, everyone here on this call can say all the tensions why it's very difficult to do wastewater for these emerging viruses in these contexts when you don't have sewer networks. And I think it's something to think about as we talk about the pressure of buzzwords in donors, right? Because all of the USAID offices want it, and they don't want to hear, well, that's actually really difficult, right? And I think we're kind of building things to try and do the best we can. And I think there's going to be more lab capacity in countries over time. And I think there's ethical issues with that I really struggle with. I mean, I don't know who's read the New York Times article. I don't want to get into did SARS-CoV start in a lab or not, but an incredibly important part in that article in the New York Times about SARS-CoV-2 and potentially starting in the lab is we have a responsibility to not spread disease via our laboratories, right? And that's incredibly important. So how do you balance that tension of not spreading disease, but localization of laboratory networks? And World Health and APHL, to answer another question, World Health and APHL have really helped. Honestly, in these countries, everybody knows every other lab. Like you go into a lab, it's not like there's a ton of labs in these countries, so you can sit down with everybody. They've all trained with one another. They've all worked together on various projects. How long from project award did it take the laboratory to be able to run PCR samples? So I would say the first 3 years of the project, there were some difficulties. I wasn't involved. I was brought in as part of the response team. And from the time I was brought in to the time we had samples was a year. And it was 12 weeks of sampling. So not so bad, but we started with HPAI and SARS-CoV-2 because it was easier. And Lhasa will have results any day now, touch wood. So I think that might, and we haven't, to answer Rochelle's other question, we haven't yet tried culture-based methods for wastewater for fecal sludge. I think that's the direction to go. I'm actually super excited about wastewater surveillance for cholera, but this particular grant spillover, again, like weirdness of donors and how money moves, it can only be viruses. We're not allowed to touch bacteria. I would love to do wastewater surveillance for cholera. I think there could be amazing work there. Cholera is exploding across countries. We've got resurgence. We're in over 30 countries now with cholera. I think there's a lot of work that should be done, but it can't be done under this grant. So I don't know if that gets at some of the tensions on this. I mean, I think in many people. Oh,
yeah,
yes.
Oh, sorry, you stole my moderating job, Danielle, but that's okay. I forgive you. You made my job easier. I have another question, which is, I feel like when it's so much work, right, for you to acquire your samples and make the measurements in country. It's way harder than field work like in California. I mean, maybe not always, but most of the time. There's all these things you've talked about and barriers and hurdles. And then what happens when you make the measurements and you sort of discover, you go to publish them, and then the reviewers are saying, like, why didn't you run a blank? Why didn't you run replicates? Why didn't you run this control or that control? Do you think that there's sort of a reduced minimal set of like quality control that we could establish for situations like this that maybe is like sub perfect but still acceptable, that sort of could help with that sort of thing? Because I can imagine that, what if you did all this work to do your Lhasa stuff and then the lab like ran out of its synthetic control or something like that? Like, what are your thoughts on that?
And I think that's I'll say probably the standards at these labs don't meet the standards of any lab or reviewer who would expect to see anything. Like in terms of what controls are being used, what's the positive control, what's the negative control? Do we have, I think that there's a question of, I think I have always been most interested in my research in these places of tension where, like, how do you balance localization versus laboratory versus safety versus testing in country? And I think those are interesting questions that require a ton of time. But yes, I can fully imagine, depending on how we when we put this paper in for publication, the reviewers will straight up come back and say, we don't think this should be published because the laboratory methods aren't confirmed enough, or this wasn't done, or this wasn't done. And I think does that mean we don't do the project? Does that mean we don't work on it? What is that? I mean, it's always kind of complicated to put country, donor, project, reviewer together.
Yeah.
Or we try and publish it as an example, a case study, as opposed to as like this is something to push forward and try to do better in the future, which is, I think, where I've landed, actually. I think we need to put
I
forward.
think it would be interesting for you to put all of that in writing in some sort of viewpoint or something at some point, Danielle. So yes, in your spare time.
Right.
Okay, so we're going to wrap up the webinar. I wish everybody an amazing summer, and thank you so much, Danielle. And Masaaki had to go because it's after midnight in Japan, and we look forward to seeing you next fall. Thank you, everybody. Thank you.
Thank you, everybody.